In this video, I want to talk a little bit about the control and regulation of the pentose phosphate pathway. Two things primarily are the rate of the actual pathway and where does G6P actually go. Okay, so let's start with the rate. So if you recall, of course, is the oxidative phase and the non-oxidative phase. Now the rate of the pathway, when we're thinking about rate, we're thinking about enzymes. And the non-oxidative phase, really, the rate at which it moves is entirely dependent on the availability of enzyme substrates. So there are those transketolases, transaldolases. Basically, if the substrates on which they act on are there, or in higher amounts or lower amounts, that's going to determine how fast this phase actually moves. So there's not really too much complexity that we're too concerned with here. But the oxidative phase, there's a little bit more to it. So if you remember, the oxidative phase was just taking glucose 6-phosphate, turning it into ribulose 5-phosphate and carbon dioxide, going from a 6-carbon molecule to a 5-carbon molecule, and creating NADPH, of course. So we've made two of those from two NADP pluses, right? And that was initially the first step in that reaction. That two-step pathway was catalyzed by G6P dehydrogenase, right? That was the enzyme that catalyzed that reaction, if you recall. Now this reaction pathway eventually releases a carbon dioxide. Now we talked about how reactions that release gases like carbon dioxide are irreversible. And if a reaction or pathway creates a gas, and that end product is sort of irreversible, then we need to make sure that we regulate this process. So this must be regulated or controlled. So that first step with glucose 6-phosphate dehydrogenase will be regulated, and the allosteric regulator of it will be NADP+. So you can consider NADP+ as an allosteric regulator of G6P dehydrogenase. Okay, so how does it work? Well, what happens when there's high levels of NADP+ and what happens when there's low levels of NADP+? The simple short answer is that high levels of NADPH, or excuse me, high levels of NADP+ will stimulate G6P, while low levels of NADP+ will inhibit. So let's actually write that in red. Inhibit. Okay, why does that make any sort of sense? Okay, so G6P dehydrogenase, one of the resulting products is NADPH. Okay, so if there's a lot of NADP+ around, that means that we have too little NADPH, right? Most of it is NADP+ and not the reduced form NADPH. So if there's too little NADPH around, we want to make more NADPH, which means you'd want to stimulate or activate this enzyme that creates it. If there's low NADP+, we want to inhibit this pathway, because that means that if there's low NADP+, that means there's too much NADPH. Most of it's in the reduced form. So we want to make less NADPH. So we want to inhibit G6P dehydrogenase. So that's a simple idea as far as the rate of the pentose phosphate pathway and how that's regulated or controlled. But now the more complex stuff, where does glucose 6-phosphate go? This is a little bit different. Okay, so where does glucose 6-phosphate go? Now, we mentioned this briefly in the first video on the pentose phosphate pathway. We said that it was need-based, so it depends on needs. And we said it really, really briefly. The needs, of course, that I want to mention are NADPH, R5P, or ribose 5-phosphate, and ATP, which is, of course, energy. We said in that first video that if ATP was needed, then the glucose 6-phosphate would go to glycolysis. And that if NADPH is needed, it would go to the pentose phosphate pathway to create that NADPH. And this is true, but it's not that simple. It's a little bit more complex than that. In fact, there are 4 scenarios that we can consider and kind of think about what's going on with this glucose 6-phosphate. Where is it going? Okay, so let's go through these scenario by scenario. First scenario, let's say we need lots of ribose 5-phosphate, but we don't really need NADPH very much. Okay, so let's consider that. First, one thing I'd like to know is, when would this be the case? So we need lots of R5P but don't really need NADPH. So R5P, we mentioned that could be used as a precursor to nucleic acid synthesis, and NADPH was more for all sorts of biosynthesis. So we need lots of R5P, but don't really need NADPH. So in this case, when would this happen? When would it happen? This is a case in which we'd need to make nucleic acids, of course, nucleic acids, and that would be highly necessary for a cell that is dividing, right? You need to replicate DNA, for instance, before a cell divides. So that's a case in which this is sort of happening. So if this is the case, if we need lots of R5P but we don't really need NADPH, what should happen or what should predominate in this case? In this case, we want to make R5P, but we don't really care about making NADPH. So which phase actually does that? Well, of course, it's the non-oxidative phase. Right? The non-oxidative phase creates R5P without creating NADPH. Okay? So let's imagine how this would sort of work. So we'd start off with G6P, and it would go to fructose 6-phosphate, and we'd create some fructose 6-phosphate. Now, fructose 6-phosphate, it can go on to fructose 1,6-bisphosphate, and in glycolysis, right? And this fructose 1,6-bisphosphate will be split, cut into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Then dihydroxyacetone phosphate will be converted into G3P. So at this point, we have fructose 6-phosphate. Let's just imagine that we have some more G6Ps creating fructose 6-phosphate. So we build up a fructose 6-phosphate. We also create a bunch of G3P. So now what happens is some of these fructose 6-phosphates and G3Ps will come together to make R5P. That, of course, these two reactions, actually let me write these in different colors. Let me put them in, let's say, purple. So this step here and this step here, these two together are, of course, the non-oxidative phase of the pentose phosphate pathway. So what we're going to be doing is we're going to have this glucose 6-phosphate, it's going to make some fructose 6-phosphates, and some of those will go on to make some G3Ps, and we're going to have two fructose 6-phosphates and one glyceraldehyde 3-phosphate each time going to make 3 ribose 5-phosphates. And that stoichiometry makes a little bit more sense if you look over here. So we're going to have 2 fructose 6-phosphates and 1 glyceraldehyde 3-phosphate coming together to make 3 ribose 5-phosphates. We take 2 6-carbon molecules, which is 12 carbons total, and 1 3-carbon molecule, which is 3 carbons total. So that's 15 carbons here, to make 3 5-carbon molecules for a total of 15 carbons on both sides. So what's actually happening to the G6P in this case? Well, if we imagine we start off with 5 G6Ps, 5 up here, right? 5 G6Ps. We're going to take those 5, which would be 30 carbons total because each G6P is 6 carbons. So we have 30 carbons to start with. And we're going to turn that into 6 ribose 5-phosphates, which is 6 5-carbon molecules, 30 carbons. So this is the stoichiometry that kind of might make this make a little bit more sense. Okay? Hope that makes sense there. So what happened here? We had the non-oxidative phase of the pentose phosphate pathway go through so that we can make some R5P without really making NADPH. Okay? So that's the first scenario. Second scenario. What if we need equal amounts of ribose 5-phosphate and NADPH? Well, okay, so we want to make R5P and NADPH. Well, there's a phase for that, right? The oxidative phase. Let me write this in white. The oxidative phase. The oxidative phase takes G6P to R5P and creates that carbon dioxide. And when it does that, of course, we get that R5P, and we also get some NADPH out of it, right? We get 2 every time that goes through. So I know I wrote equal amounts up here, and each one of these steps will make 1 ribose 5-phosphate and 2 NADPH. But essentially the idea here is that we're making NADPH and R5P both at the same time. So we're making 2 NADPHs for every 1 R5P, but it's not too different if you think about it. Okay, but the point is we're making both in this scenario. What about the third scenario? This third scenario here, what if we need much more NADPH than, what if we need much more NADPH than we need R5P? Okay, So when would this be the case? We want a lot of NADPH, but we don't really care about making R5P. Okay. When would this be the case? An example of when this would happen is in the liver, right? Our liver needs lots of NADPH because it makes a lot of fatty acids. And for that biosynthesis, right, we need a lot of reducing power. And R5P is not really necessary in that case. You just need a lot of NADPH. So if this is the case, what should actually happen? Well, we don't actually have a phase for this, right? We don't have a phase that just creates NADPH and, without making R5P. The oxidative phase makes both. The non-oxidative phase makes only R5P. So we don't have something that makes just NADPH. What would happen here? This is a little bit tricky. Okay, this is kind of tricky. But what are we doing? Basically what we're doing here is we're taking one glucose 6-phosphate, okay, which is a 6-carbon molecule, and what we're doing with that is we're going to fully oxidize it to make it into actually 6 carbon dioxides. So we're taking one 6-carbon molecule and fully oxidizing it. I'll put that here. Oxidizing it fully to 6 carbon dioxides. Okay, so how do we do that? Okay, so this is a little bit tricky, but try to bear with me for this. Okay, so what we want here is only the product of the oxidative phase, right? We just want that product, the NADPH, but we don't want the RU5P product. Okay, so how would this work? Well, one thing for sure is that we're going to take this G6P and convert it into RU5P and get some NADPH out of it. Okay, so that's definitely true. Okay, we're going to get this. We're going to get 2 NADPHs here from this oxidative phase. Okay, and of course we're going to knock off a carbon dioxide, right? So I'll put that maybe in green. Okay, so we're going to also get a carbon dioxide out of each one of those steps. Okay, but then what happens? So this part here is the oxidative phase. Then we're going to take this RU5P, but we don't want this, right? We don't care for this, especially because immediately it's going to actually be turned into R5P. But we don't want this either, right? We don't care to have this. So what we're going to do is we're going to actually have this go to G3P and to F6P via actually, let me put that in purple, those arrows, just to kind of stay consistent with the fact that I used them for the non-oxidative phase earlier, right? So now we're going to go backwards, right, in the non-oxidative phase. So here, this is the this is the non-oxidative phase, right? We're taking R5P and going back to G3P and F6P. So what happens then? So now we're going to go back and basically go through gluconeogenesis. We're going to take this G3P and this DHAP and bring them back together to make fructose 1,6-bisphosphate, and then to make fructose 6-phosphate, and then we're going to have the fructose 6-phosphate go back to G6P. So this whole process here is, of course, gluconeogenesis, right? We're going to have gluconeogenesis going on. We're going to have the non-oxidative phase, and we're going to have the oxidative phase up here, right? So there's a lot going on in this one, okay? But what ends up happening in terms of the stoichiometry? So let's say we started off with 6 glucose 6-phosphates. So we have 6 times 6 carbons, 36 carbons to begin with. We want NADPH out of this for sure. So we go ahead and we take all these glucose 6-phosphates, turn them into 6 ribose 5-phosphates, and we make a bunch of NADPH. Cool. We're also making these carbon dioxides here. So these 36 carbons end up 30 of them remain in these 6 5-carbon molecules of R5P, and 6 of them, of course, are released as carbon dioxides. Then we take these R5Ps, which were right here, right, and we're going to have them go in the non-oxidative phase, right? So actually, it might be helpful to number this. Maybe number 1 would be here, right? That first step. The second step, or the second sort of pathway or idea that we're thinking about is the non-oxidative phase, which we have here, and then the third one, we could have this gluconeogenesis going on there. Okay, so the second thing, we have ribose 5-phosphate. We have 6 of them that we created here, and they're going to go through the non-oxidative phase, right? And they're going to, so we've seen before that 3 ribose 5-phosphates make 2 fructose 6-phosphates and 1 glyceraldehyde 3-phosphates. Well, if we go from 3 ribose 5-phosphates to 6, we're doubling that number there, so we double the numbers for fructose 6-phosphate and glyceraldehyde 3-phosphate as well. So we start off with 30 carbons here, then we have 24 here and 6 here, okay, which is still 30. Now what happens? Well, now these fructose 6-phosphates and glyceraldehyde 3-phosphates, these are, of course, the glycolytic intermediates, glycolytic or gluconeogenic intermediates. Now, what they're going to do is go through gluconeogenesis and create some glucose 6-phosphates. So we go from 24 carbons here, 6 carbons here, and then we're actually going to make 5 glucose 6-phosphates. If we have 30 carbons total, that would equal 5 glucose 6-phosphates. So if we go through and calculate the overall reaction, we can sort of cross out anything that's on both sides of these arrows here. So we created 6 ribose 5-phosphates, and then we used them up. We created 4 fructose 6-phosphates, used them up. 2 glyceraldehyde 3-phosphates, used them up. So all that's left is we have 6 glucose 6-phosphates there, and 5 here, right? And then these 6 carbon dioxides. But with these glucose 6-phosphates, right, these 5 can get crossed out, and then we cross out just 6 of those, right, and we're left with 1. So we go from 1 glucose 6-phosphate to 6 carbon dioxides. So the overall reaction is 1 glucose 6-phosphate, 6 carbons, to 6 carbon dioxides, and we create a bunch of NADPH, and effectively don't create any R5P. Okay, so that's that third scenario. The last scenario is if we need NADPH and ATP. So what should happen and predominate here? Okay, so we want to create NADPH and ATP. So we want the phase that creates NADPH is the oxidative phase, and the thing that creates ATP is, of course, glycolysis. So we want those two things to happen. Okay, so the oxidative phase is, of course, turning glucose 6-phosphate into ribose 5-phosphate, and then we'll get some NADPHs, excuse me, 2 NADPHs out of this, and then 2 NADP pluses go in. And then, of course, we get some carbon dioxide. Oops, not purple. One green. So we get some carbon dioxide out of there too. Okay, so that's the oxidative phase, and we want glycolysis to occur. So what we're going to do is we're going to take any of these RU5Ps that are made, turn them into R5Ps. Now these R5Ps can go through, make some fructose 6-phosphates and some glyceraldehyde 3-phosphates. Now those things will go through through glycolysis, of course, right? So again, I need to make sure I'm consistent with these colors. So this is the non-oxidative phase here, right? Going through to make those, and then what we have is glycolysis happening. So now fructose 6-phosphate will go down to fructose 1,6-bisphosphate. This will be cut into DHAP and G3P. DHAP will be turned to G3P. So now we have a buildup of G3P. Now those G3Ps can be oxidized, turned into pyruvate, and when that happens in glycolysis, we're going to get 2 ATPs for every G3P that we have. Okay, so basically what happens is the R5P that's made here is taken to glycolysis essentially, right, via the non-oxidative phase to be used for energy. Okay, so now we're getting 2 ATPs for every G3P that we get here. Okay, and then of course once we have this pyruvate, that pyruvate can be further oxidized for more ATP or using a biosynthetic pathway, whatever is necessary. So as far as the stoichiometry goes here, we have, let's just imagine we have 3 glucose 6-phosphates, so that would be 18 carbons total. And we turn that into, if each one of these goes through, we get 6 NADPH and 6 NADP pluses going in to make 6 NADPHs, right? 2 for each of the glucose 6-phosphates. And then we create 3 carbon dioxides, and we have 5 pyruvates. So if we ended up creating 5 pyruvates, that means we must have gotten 5 NADHs made. Okay, so we go from 18 carbons here to 18 carbons here. And what is the actual amount of ATP that we get, right? How much ATP do we actually get? Well, if we think about it, these 3 G6Ps made 3 R5Ps, right? 3 R5Ps, we keep coming back to that number, makes 2 fructose 6-phosphates and 1 glyceraldehyde 3-phosphate. each glyceraldehyde 3 phosphate gives us pretty much 2 ATP. So we have 2 ATP here from that one glyceraldehyde 3 phosphate, and these two fructose 6 phosphates, when they go through, that gets converted to fructose 1,6 bisphosphate, and that is split into DHAP and G3P. DHAP, it has to be converted into G3P. So essentially, each one of these fructose 6 phosphates gives us 2 G3Ps. So 2 G3Ps times 2 fructose 6 phosphates is 4 G3Ps, right? And 4 G3Ps times 2 ATP each will give us 8 ATP. So we basically get around, so we get around 10 ATP total out of this fourth scenario. Hope that video was helpful. Thank you for watching. Hey folks, my name is Mafood. I am a tutor. I graduated from UC Riverside with a degree in biology, and on graduation day I had a little bit of trouble with the wind, as you can tell. The subjects I tutored for are listed here. In the description below you'll find my email address if you have any questions, and you'll also find the links to 2 listings, my university tutor listing, which is for in-person tutoring, and my Google Helpouts listing, which is for online tutoring. Thanks for watching.